Hello, students. Welcome to my class. I'm Keerti, and today we are going to study about principles of inheritance and variation. Under this chapter name, we are going to discuss about Mendel's law and deviation. Right? Before moving into the chapter, let's first decode the title of this chapter, which says Principles of Inheritance and Variation. Right? Principles means there is some kind of rule, okay, some kind of way that a particular thing is happening, something that is happening in a continuous manner. And these principles are concerned with the inheritance. Okay. Inheritance means something, some character that is being transferred from parent to offspring. What do we inherit, right? We always say when a newborn is born, you have always seen that your grandmother, grandaunt telling everyone, oh, see this child is happened to be like his father, his eyes belongs to his mother, right? So these are what, some kind of character that the newborn baby must be inherited from their parents, right? But the child, the newly born child, is not exactly same as their parents, right? There is some variation. Even in siblings, even if we as a siblings, we are different from each other. If a couple is having three child, all the three child will be different from each other. So how this process, how this pattern is being governed, this chapter is all about this only, how what kind of principle is there that this inheritance is taking place in this manner, right? So let's start our chapter. We have already studied that genetics is the study of inheritance and variation of characters. Character may be anything you can think of, like the skin color, the hair length, the type of texture of hair, okay, the height that you are talking about, anything, right? So Gregor Mendel, who is known as the father of genetics, established some basic laws that is governing the inheritance patterns. So his experiment earlier was based on pea plants, right? He has performed n number of experiment in this peas plant and ultimately derived some basic laws, right? He was not aware at that time that what were the factors that he called, right, that are governing the process of inheritance. But many scientists followed his path. And in today's modern biology, we already know that these factors are known as genes. So these genes are responsible for the characters, for any character that you can think of, right? So let's start how the Mendel started his early experiment. Okay, Mendel conducted various hybridization experiments on pea plants, okay, using true breeding lines. And in total of 7 characters were studied by him. The characters that we are talking about. All the characters were visibly present. One can visibly see that there were these 7 characters present in the pea plants. These 7 characters were the color of flower, the form and color of seed, right? The pod shape and color. Okay. If the pod has, smooth shape or it has some wrinkles on it, it is constricted in shape. If the pod has yellow color or green color, right? The color of the flower, that is also visible. Whether the flower will have a white color or it will have a violet color. Okay. And after, that the seed that is formed from the plants, whether they are round or wrinkled. Okay. The cotyledons present inside the seed, whether they are yellow or green in color. And the stem length, whether the stem is taller or shorter, right? And the pods, the placing of pods, whether it is axial or not, whether it is terminal, present at the end, or it is present at the center. So in total, there were 7 characters that he talked about. Okay. Earlier, he was taking into account only one character of contrasting trait, right? Only one character that is contrasting in nature. So that cross is known as monohybrid cross. It means he considered only one character at a time. So his first experiment was based on the height of the plant, the tall and dwarf plant. As the name suggests, tall means tall in nature. It has more height, and dwarf means short in nature. Okay. So he take one tall plant and one dwarf plant, right? He made a cross, right? And what he sees in the first generation, that all the plants were dwarf, right? All the plants were tall. Sorry, my fault, right? What he next done, he crossed this tall plant. He crossed this tall plant. It means he self-fertilized the plants. Okay, the self-pollination has happened. And again, he has seen that these generations show a phenetic ratio of 3:1. It means there were two type of plants seen. Okay, tall plant and dwarf plant. Okay, so this generation is known as F1 generation, when the cross is taking place between the parents. F1, first filial generation, right? And when this are, again self pollination take place, this will produce another generation known as F2 generation, or second filial generation. So the phenotypic character it shows was tall as well as dwarf, right? So on the basis of this, he derived 3 laws. First law is law of dominance, right? Law of dominance says that the characters are controlled by discrete unit called factors. Because he was not aware of the term genes. Later, the other scientists discovered that these factors were actually the genes that he was talking about at that time, right? And these factors occur in pairs. Pairs means there will be two forms of a single factor, and one factor dominates the other. And here this term of alleles is coined, right? So if we are taking into account the height of the plant, say height of the plant, and we are symbolizing it with letter T. So for height, this gene has two forms, two allelic forms, T and T. And this capital T actually dominates the small t. It means whenever this capital T is present, the small t will not be able to express itself, right? Okay. So what was happening when the cross he performed between tall plants, which ultimately lead to the formation of tall and dwarf plant in the F2 generation. This happens because these tall plants were not homozygous. The genotypic constituent of this tall plant was not homozygous. It means it must be present in a heterozygous form. This is known as heterozygous form. It means both the alleles of the same gene are not identical, right? That is why when this F2 generation is seen with two kind of plants, tall and dwarf, in a ratio of 3:1. Right? We will study about this in a more elaborate way. Next law was law of segregation. This law of segregation states that alleles do not blend, do not get mixed, right? This will segregate during the gamete formation, right? It's not like that, that these two alleles are gotten mixed, right? So this segregation will take place during gamete formation, and gamete will receive only one allele of a gene pair. Suppose if this is the parent and this is going to form the gamete. Okay, okay. So only one allele is going to be placed in a single gamete, right? And third law was law of independent assortment. Independent means it is not dependent on one another. So when two pair of traits are combined in a hybrid, the segregation of one pair of character is independent of the other, right? So three laws were given: law of dominant, law of segregation, law of independent assortment. Dominant means, of course, the one allele is dependent on, dominant over the other allele. Next is segregation. Only one allele of a gene pair will get into the gamete. And third is, if more than one pair of character is considered, or two pair or three pair, these will be assorted independently. It will not happen that if a plant is tall, so if it is having a white flower, so in the next generation only the tall plant will have the white flower. That will not be the case. It may happen that the small heighted plant will also have a white flower. Okay, so the assortment will be independently, right? This graphical representation of the genotype of the plant forming the gametes and the resulting of the offspring is done in a very stepwise manner, and in the form of Punnett square, in a form of square having many grids, where the plant, where we can generate the type of gametes from the two plants. Okay, whatever type of gametes is generated, now we can make a cross and produce the final offspring, right? So it's a representation technique where you can easily make the gametes and cross them to see the resulting pattern, right? This was developed by Reginald C. Punnett. To honor him, it is called as Punnett square, right? This incomplete dominance shows that the dominance is there, but in an incomplete manner. Okay, it means that neither of the allele is completely dominant. For example, this is the cross that is given. Suppose this R is dominant, of course, it is homozygous. Now this pair is homozygous. This is also homozygous. If this flower is red in color and this small r is white in color, so according to Mendel's law of dominance, the F1 generation must have a combination of this, and all plants should be red in color, right? But in actual, it is not happening. This R are actually forming a flower which is pink in color. What does it implies? It implies that this capital R is not in a dominant form. So there is some expression of this R allele and there is also some expression of this white allele. That is why the F1 generation plant here all are pink in color. A third color is formed, right? So this process, this cross leads to leads to another term, that is incomplete dominance. And next come the codominance. What is codominance? Right? This codominance is the classic example is the human ABO blood group system. Okay. We already know we have four types of blood in case of human blood group, right? A, B, AB, and O, right? So, in case of blood group where the blood group is AB, both the alleles IA and IB are present, which is known as AB blood group. If the person is having AB blood group, it means both the alleles are expressed equally. That is known as codominant, right? So our next is what is multiple allelism. This can be also explained through the ABO blood group system, where we not only have IA alleles, IB alleles, it means this I gene is controlling, right? We also have a small I, right? When we are going to have O blood group of the individual, actually this small I is present in those individuals. So here we can see not only two allelic forms are there, actually there are three allelic forms. And that is known as multiple allelism, right? Next is our chromosomal theory of inheritance. This was proposed by Sutton and Boveri. It states that genes are present on chromosome and their segregation follows Mendelian laws, right? So in modern times, you already know this chromosome is made up of DNA and a particular segment of DNA is known as genes, right? And this genes segregation, right? The assortment, the segregation is governed according to the Mendelian laws, right? There is another term known as linkage and recombination. This was discovered by scientist named Hunt Morgan, which performed his experiments in Drosophila, right? So he has performed various experiments and concluded that genes on the same chromosome are inherited together unless recombination occurs, right? So this we will study. You don't have to worry. We will study what kind of genes he was talking about, what kind of characters he studied. Okay. Another term is there known as polygenic inheritance. That is, the traits are controlled by several genes. Earlier, we have seen that only single gene is controlling a single character, but now we know that traits are controlled by several genes. For example, human skin color and height. This we will study through how, when we are going to study about the traits, we will consider how these alleles are actually working, right? Okay. So we will study each and every Punnett square that is being discussed by Mendel. Okay. I hope I'm clear with this overview of this topic. We are going to study in deep and detail regarding this Mendel law when we are going to study the dihybrid crosses, when more than one character is being considered, how this independent assortment is actually taking place. But beforehand, you must have an idea what is segregation, what is independent assortment. A little bit of idea is enough for going into that deep of the chapter. So we have come to an end to this session. If you have any doubt, you can contact me through my Urban Pro profile. Till then, you can go through the chapter, collect your doubts, and come back again when we will meet in our next session and we will study further. Right? Okay. So till then, take care. Thank you for the session. Thank you.